when we're soil sampling, when we're thinking about taking a sample from our paddock, we need to consider the size of the paddock. Here today we've got quite a small paddock and a small bay within this paddock that we're going to test. And in this paddock we're going to just do a straight line of samples. So we're going to take up to 20 cores in our soil corer, which will give us about a kilo of soil sample that then we can send off to the lab. So when we're trying to take a soil sample, we're trying to get a representative view of that paddock. So we don't want to be sampling and plunging our soil core into things that are going to contaminate our sample, like for instance manures or anywhere where the cattle have been camping where there's going to be excess urine deposits, and also not in the row where perhaps there's going to be fertiliser. So we won't be sampling in the middle of the row, we won't be sampling in the row, we'll be sampling in the middle of the row to reduce that contamination. And we want to think about how we travel across this paddock and take these samples. So in the soil sampling handout that you would have received on the day, there's a breakdown of different patterns that you can use. For instance, you can choose to do a direct line and take samples at an even space, like maybe every 10 metres, or you can choose to do a zigzag pattern. So for instance, walking for 10 metres, taking a sample, and then heading an opposite direction for 10 metres, take a sample, head the other direction, take for 10 metres and take a sample, so that we end up with a crisscross pattern. You can divide the paddock into quadrants, so break it up into blocks and take a sample from each block. It might be a 20 by 20 metre area, and you take one sample in each until you've sampled the whole paddock. But today we're just going to do the straight line, and we're going to go in the middle of the row for the full length of the paddock, and we'll take about 20 or 25 subsamples. The other thing to remember when sampling is most of the time we're using a plastic bucket to put our samples in, so that we're not getting any contamination from any metal buckets that may have may distort the sample when we get that back. So once you've taken your sample and you have your bucket of soil samples, you then want to get some sort of set of scales or kitchen scales or something, and tip your soil sample into a paper bag, and then weigh it, and you want to have about 400 grams to be sent off. So I like to double bag my samples, so I put them in a paper bag and then fold them up into a square of about this size, and then I double bag it in another paper bag, and then bag it into a Glad or one of those Ziploc plastic bag, and then you put your details on the front of the plastic bag. When you're taking your sample, you need to remember that we'd be sending the sample express post, and it usually goes up to Soil Food Web and Environmental Analysis Labs in Lismore in New South Wales. So when we're taking the sample, we need to consider taking it either on a Sunday evening or a Monday morning or Tuesday morning, and sending the sample off that day in the express post, so it doesn't sit in the post over the weekend. And if we have to take it a day before, for instance, and we can't send it off until the next day, it's a good idea to refrigerate the sample, just to prevent any humidity changing the sample. So always take it on a Sunday, Monday or a Tuesday and send it off that day. The other thing that you'll notice in the handout sheets is there's a specific way of labelling the bags, which I like my clients to follow. So if you are going to take a soil sample, in your handout sheets it'll tell you that you need to have your name and paddock name, as well as your phone number or my phone number, so that the sample can be identified, and it needs to go with that form in the express post bag, so that when it gets to the lab, so that they know whose sample it is and who to send the results to. So usually I'll put my details on there, so the results be sent back to me for interpretation before it then goes back to whoever has ordered the soil sample, and the results will come back through me and then go back to you. We've identified our path that we want to sample down, so just pop the soil core in here, give it some pressure. Okay, so sampling to that sort of depth, we're going to get about 10 to 12 centimetres of soil core. And as you can see, this one has an open window here so we can see our sample. Just borrow that screwdriver. Sometimes if it's quite sticky, you might need to give it some assistance to come out. And one thing we want to remember when taking a biological sample is that we like to get some of the root material of the plant, the live root material, to be sent off to be analysed for mycorrhizal fungi associations on the root. If we don't have that live root material in our soil cores, then we're not going to be able to get that reading back on our biological testing. And it is different for nutrient sampling. We don't want to have too much organic matter going in here that's going to elevate the organic matter percentages in our samples. So when we're bagging off our soil for going into our plastic bags and then into the post, we want to just make sure that the portion that we put in for the biological sample has some live roots, and that the nutrient sample is free from any extra organic matter that might be on the soil surface that's not actually incorporated into that soil core.